Imagine your body's clock just, well, stuck on fast forward, speeding through life stages way faster than normal. That sounds like science fiction, maybe.
But it's actually real for people with certain uh, very rare genetic disorders. It gives this appearance of premature aging.
Yeah, and these conditions, they're generally grouped together as progeroid syndromes.
That's the term. They show symptoms that look a lot like normal aging, but everything happens much, much earlier and progresses really rapidly. It gives us this, well, unique though difficult insight into how aging actually works.
And for this deep dive, we're looking at a really interesting research paper. It zooms right in on one specific aspect, one of the key things scientists track with aging telomere length.
Ah, yes, telomeres.
You can think of them as like little protective caps on the ends of our chromosomes, a bit like the plastic bits on shoelaces,
Yeah,
stopping them from fraying.
good analogy. And as we age, those caps naturally get shorter. It's a process called telomere attrition.
So scientists have kind of wondered for a while if this shortening, this attrition, might be a common factor in lots of these progeroid syndromes, especially the ones where DNA repair seems to be faulty.
Exactly. But it wasn't totally clear just how common it really was across, the whole range of these conditions, because they are quite diverse.
So that's our mission today for this deep dive. We'll unpack what this study found. We want to understand which of these specific premature aging syndromes do show significant telomere shortening, which ones maybe don't.
And what that difference actually tells us,
Hmm.
both about the underlying causes of these rare syndromes and maybe even about normal aging too.
Plus, there's a really fascinating side finding in the paper we need to touch on. It's about certain genetic variants, ones known to be sort of protective against common age-related diseases, and if they have any link to telomere length.
Yeah, that's an interesting twist.
And the researchers here, they used a pretty smart method, mostly estimating telomere length using DNA methylation data, those chemical tags on DNA.
Right, epigenetic data. And they did back up some findings with another technique, qPCR, for validation.
Okay, let's dive in then. These progeroid syndromes, they have a history, right? They weren't always called that.
No, exactly. Conditions that look like premature aging, something like xeroderma pigmentosum, which affected the skin's ability to repair UV damage, that was described way back, late 1800s.
Yeah.
It's
But
54, I think.
wow. And the actual word progeria?
That came later, 1904. It's from Greek, meaning prematurely old. Originally, it was used for what we now call Hutchinson-Gilford progeria syndrome, or HGPS, the classic childhood form.
And then progeroid, meaning like progeria?
That term popped up in 1978
Hmm.
for conditions that had similar features but weren't quite identical to HTPS. And they really are rare and very heterogeneous. Lots of different combinations of aging signs.
Mostly caused by just single errors and single genes disrupting specific cell functions.
Right. And the paper groups them broadly by what's going wrong. Sometimes it's the nuclear envelope, the membrane around the cell's nucleus. That's like HTPS or mandibuloacral dysplasia. Or the issue could be defective DNA repair. That leads to genomic instability, basically the DNA getting damaged more easily. Think Werner syndrome, Bloom syndrome, those kinds of conditions.
And crucially, some are specifically telomere problems, right? Telomeropathies.
Yes, exactly. Dyskeratosis congenita, DKC, is the textbook example there, often caused by mutations in genes whose whole job is maintaining telomeres, keeping them long enough.
So even though they all look like accelerated aging, the why behind it isn't always the same mechanism as normal aging.
Precisely. But by studying what breaks in these syndromes, we can sometimes find new genes or pathways that are actually relevant to how everyone ages. It's a valuable, if indirect, approach.
Which brings us right back to telomeres. He said they're dynamic structures protecting the chromosome ends.
Protecting the important genetic information during cell division.
And this shortening telomere attrition, it happens because the copying machinery, DNA polymerase, it can't quite get to the very, very end of the chromosome each time a cell divides.
Right, the end replication problem.
Right.
And most of our normal body cells, our somatic cells, they don't have much telomerase. That's the special enzyme that can add DNA back onto the ends. So they just gradually shorten over our lifespan.
And as you mentioned, some earlier studies had suggested telomere shortening in specific progeroid syndromes, like Nijmegen breakage syndrome, even HGPS in certain cell types like fibroblasts.
Which led to this hypothesis. Maybe shorter telomeres are a common feature, especially if DNA repair is faulty in the syndrome.
So this paper aimed to really test that. How common is it across a broader range? What did they find overall?
Well, first they look at all the progeroid syndrome patients grouped together
Hmm.
compared to controls, using that main DNA methylation method.
The DNMT latch.
Right. They did find significant telomere attrition overall for the combined progeroid group.
Okay, so initially it looks like the hypothesis might hold up, but you said it gets more nuanced.
It really does, because when they broke it down and looked at each syndrome individually, the picture changed quite a bit. Significant shortening was definitely there for some, but clearly not for others.
Let's get into specifics then. Which ones did show significant telomere shortening?
Okay, first up, classical Werner syndrome, WS. Clear, significant telomere attrition here. And this was seen with both the DNA methylation estimation and the qPCR validation.
And classical WS, remind us.
That's the adult onset type. Features like early graying, hair loss, skin thinning, cataracts, diabetes, cancers, basically age-related diseases hitting much earlier. It's caused by a faulty WRN gene.
And the WRN protein is involved with telomeres.
Directly, it helps unwind DNA structures, including its telomeres, and works with the proteins that protect the telomere ends, the Sheltern complex. The mutation messes that up. So finding shorter telomeres makes perfect biological sense here, and it strongly supports the idea that telomere problems drive the aging in WS.
Okay, so that one fixed the telomere hypothesis nicely. What else?
Next was Berardinelli-Seep congenital lipodystrophy type 2, CGL2. This also showed significant telomere attrition with the DNA methylation method.
CGL2, that's a lipodystrophy, right? Problems with fat storage.
Exactly. Caused by variants in the BSCL2 gene. Now, the paper notes the qPCR validation trend was weaker here, but they also mentioned the sample size for that test was tiny, just 3 patients.
So maybe not enough power for the qPCR.
Possibly. But the DNA methylation finding is interesting because this study seems to be the first to report telomere shortening in CGL2. It raises new questions about how the BSCL2 gene might, perhaps indirectly, affect telomere length, something we didn't really know before.
Okay, a new potential link there. And the third one with shortening.
Disc keratosis congenita, DKC. And frankly, this wasn't a surprise at all.
Because DKC is a telomeropathy, right? Short telomeres are sort of its defining feature.
Pretty much. It's often caused by mutations in genes directly involved in making or regulating telomerase, the enzyme that lengthens telomeres. So less telomerase activity leads to shorter telomeres. Measuring telomere length is even used diagnostically for DKC sometimes.
So the study's findings here just confirm what was already well established using other methods.
Exactly. It lines up perfectly with older studies using techniques like FlowFesh or qPCR. And the paper even mentions how, within DKC, the severity of shortening can depend on which gene is affected, or even where the mutation is in the gene. Some variants seem to cause shorter telomeres than others.
Okay, so classical WS, CGL2, and DKC show the significant shortening. Now, the really interesting part, perhaps, which syndromes did not show significant telomere attrition in this particular study?
Right. This is where it challenges the idea that it's a universal feature. First on this list, Hutchinson-Gilford progeria syndrome, HGPS.
The classic childhood progeria.
Yes. Using their DNA methylation analysis from blood samples, they found no significant telomere attrition.
But wait, I thought HGPS was linked to telomere problems in some studies.
You're right to remember that. This is a really key point of context. Previous work did show telomere issues in HGPS, but usually when looking at fibroblasts, those connective tissue cells that are very affected in HGPS, they produce the faulty progerin protein.
Okay.
But the paper cites another important study which found that in blood cells, specifically hematopoietic cells like T cells, B cells, NK cells from HGPS patients, the telomere length was actually normal.
Ah, so this study looked at blood and found normal length, which matches that specific finding in blood cells.
Exactly. It's consistent. And the likely reason is that the LMNA gene, where the HGPS mutation occurs, isn't expressed much, or maybe at all, in those blood cell lineages, whereas it's highly expressed in fibroblasts. So the cell type you test really, really matters.
Fascinating distinction. Okay, who else didn't show significant shortening?
Atypical Werner syndrome, WS. This is where patients have Werner-like features, but don't have the typical WRN gene mutation. It's genetically diverse, could be mutations in LMNA, PLLD1, CESBRTN, SAMHD1, various genes.
And the lack of shortening makes sense, given that mix of genes.
It seems to. While some, like LMNA, can potentially affect proteins linked to telomeres, others like PLD1 or SBRTN are more involved in general DNA replication or repair without a known direct role in maintaining telomere length specifically. So finding no consistent shortening across this mixed bag of genetic causes kind of fits.
Okay, and there were 2 others with no significant finding.
Yes, cerebroretinal microangiopathy with calcifications and cysts, CRMCC, and Wiedemann-Rottenstrauss syndrome, WRS.
Now, CRMCC, isn't that also considered a telomeropathy caused by a variant in the CTC1 gene involved in telomere replication?
It is. So you'd absolutely expect telomere defects.
Yeah.
The study basically says the reason they didn't find a statistically significant difference here is almost certainly the tiny sample size. They only had data from 2 CRMCC patients.
Just 2. Okay, can't draw conclusions from that.
Exactly. Same issue with WRS, which is a neonatal progeroid syndrome, also genetically varied. They didn't find significant attrition, which they note is consistent with one previous analysis on WRS fibroblasts, but again, N equals 2. Sample size is just too small.
So putting it all together, the big picture is that telomere shortening is definitely not a universal thing across all these syndromes that look like rapid aging.
That's the core conclusion. This variability really hammers home that these conditions have different root causes, different molecular pathways going wrong. They might share some outward signs, but they aren't all driven by the same underlying aging clock mechanism, if you will.
And the implications for treatment seem huge, right? If you're developing a therapy to boost telomere length.
It might be great for DKC or maybe classical WS, but it likely wouldn't do much for HGPS, at least based on blood cells, or maybe atypical WS, because telomeres aren't the main issue there. You need targeted approaches based on the specific defect.
Tailored medicine for these rare conditions.
Absolutely. That diversity is key.
Okay, that covers the progeroid syndromes themselves. Now, let's switch gears slightly to that other part of the study, the protective variants.
Right, looking at the potential upside, genetically speaking. They wanted to see if certain genetic variants known to lower the risk for common age-related diseases had any connection to telomere length.
Which ones did they look at specifically?
They focused on the APOE2 allele that's linked to a lower risk of Alzheimer's disease, and also specific loss-of-function variants in PCSK9 and APOC3. These are tied to lower cholesterol levels and reduced risk of heart disease, atherosclerosis.
And they compared people with these variants to
controls carrying the standard APOE3 allele.
So did having one of these protective variants associate with longer telomeres compared to the controls?
Interestingly, no. When they looked at each protective variant individually, or even grouped by gene, APOE or PCSK9 APOC3 together, there wasn't a statistically significant difference in telomere length compared to the APOE3 controls.
Huh. Okay. So individually, no strong signal against regular controls, but I sense a but coming?
There is a but. The really significant finding emerged when they compared the combined group of people carrying any of these protective variants against the combined group of patients with progeroid syndromes.
Uh-huh. And what happened then?
Then they saw a significant difference. The group with the protective variants had significantly longer telomeres than the group with progeroid syndromes.
Wow. Okay. So the contrast is really between the protected group and the accelerated aging group. That's quite telling.
It is. One group genetically predisposed to faster aging features, some with shorter telomeres. The other group genetically predisposed to avoiding common age-related diseases, and they tend to have longer telomeres, at least compared to the progeroid group.
Did the protective group also have longer telomeres than the general controls?
Slightly longer, yes, compared to their main control group who were older than 10. But that specific comparison didn't quite reach statistical significance in this analysis. The big difference was against the progeroid patients.
It definitely suggests some kind of link, though. Maybe these protective variants help maintain telomere length better over time or protect against factors that shorten them. And the paper mentioned these same variants were also linked to slower epigenetic aging.
Yes, in another recent study by some of the same authors. So it hints at a broader connection to biological aging markers. But the specific mechanisms by which these protective variants might influence telomere length, that's still an open question and needs more research.
Definitely an area to watch. Okay, let's quickly recap the methods and the study's own acknowledged limitations before we wrap up.
Sure. So the main tool was that DNA methylation-based estimator calculating DM-TILI-DAG telomere length adjusted for age and sex. They used qPCR to validate some key findings.
And the strength of the methylation method is using existing large data sets, right, from blood samples.
Exactly. It leverages those big public resources. But limitations are important too.
Like pulling data from many different sources, heterogeneity.
Yes, that's one. And the big one we already discussed, small sample sizes for some of the really rare syndromes, like CRMCC and WRS with only 2 patients each. You just can't get strong statistical power with numbers that low. It's the reality of studying diseases this rare.
Yeah.
DKC is maybe 1 in a million, HGPS 1 in 4 to 8 million, CGL2 maybe 1 in 10 million.
Yeah, finding enough patients is a huge challenge.
Absolutely. And the final point they mentioned was that this DNA methylation estimator might not be super precise at measuring the very shortest telomeres within a cell.
And those are thought to be the really critical ones.
Potentially, yes. It's often thought that reaching a certain threshold of critically short telomeres is what triggers cells to stop dividing, to enter senescence. So while the estimator gives a good average picture, it might miss some nuance about those few really short, potentially problematic telomeres.
Okay, good to keep in mind. Sort of a forest level view, maybe not every single tree.
That's a reasonable way to put it.
So wrapping up this deep dive, the really big takeaway seems to be this confirmation that telomere attrition, while definitely a feature in some accelerated aging syndromes like classical WS, CGL2, and DKC.
It's clearly not a feature in others, like HGPS and blood cells, atypical WS, and potentially CRMCC and WRS. Those sample sizes were limited there.
Highlighting that these progeroid syndromes really do have diverse underlying causes. Different paths can lead to features that look like rapid aging.
Exactly. And then there's that intriguing hint about the protective genetic variants, the APOE2, the PCSK9, APOC3 loss of function ones. They seem associated with longer telomeres, especially when you contrast them with the progeroid group.
It feels like studying these incredibly rare conditions is like finding these, edge pieces of the aging puzzle. Each one shows you something different about how the whole system works or how it can break down.
Well said. Seeing these extreme examples really does illuminate the complexities of normal aging for all of us, how it proceeds, and potentially how it might be protected.
Which leaves us, and you listening, with something to think about. If telomere shortening isn't the common thread linking all these different types of accelerated aging, what other fundamental aging mechanisms might be more universal? What else is going wrong across the board?
And on the flip side, if these protective genes really are linked to maintaining longer telomeres, as this study suggests, could figuring out how they do that give us new clues, maybe even new strategies for understanding and perhaps influencing typical healthy aging down the road?